that, I mean, one of the hot areas these days is what are called organoids. And so for the brain, they call them cerebral organoids. And what people have done is directed embryonic stem cells that can make any tissue to make brain tissue in particular. And then these cells self-organize into what looks like mini brains, small little brains. And you can do this with human cells as well as in mouse cells. But nobody's going to transplant a mini brain into a regular person's brain. It's too
Yeah.
uncontrolled right now. But you can see that there's a certain amount of self-organization that's really encouraging because you turn them in, you take some embryonic stem cells, turn them into brain stem cells, get them to make a whole bunch of different types of brain cells, and then they sort of self-organize. And so the fact that they self-organize into what are called cerebral organoids sort of leaves people with hope that maybe in vivo, if we stimulate the stem cells that are present in the brain, then
Yeah.
maybe they'll self-organize the hippocampus so that it works better and can store new memories. But that's more of a hope right now than a demonstrated fact.
Yeah, that's interesting. Being able to create like brains in like a petri dish, like very yeah no like I don't even know because I think part of the problem too is like we have a like poor understanding of how the brain functions now. That to like create one and then have that be someone's brain, like we would have no idea what like personality they would have. We'd have no idea like what like how that person would act.
Well, and especially
So,
if you're trying to replace cells in a memory part of the brain, what happens to the
oh
old
wow,
ones? They're
yeah.
gone. Do you get new memories? It's sort of a joke, but yeah.
Yeah, yeah. I mean, like it is, but like if you could create brains like that, then like yeah, then in theory you could understand. I mean, it would help you understand how the brain works.
Yeah, I mean, you don't really know how the brain develops until you can do it yourself in a dish, right?
Yeah,
Otherwise, it's just speculation that we think this cell gives rise to this cell. It becomes connected with another cell type. We don't really know that, right? It's a hypothesis, but you hope that these mini brain organoids, do have some self-organization ability, and so you may not have to program everything in. You can let some of the cells self-organize, and that's sort of a hope of a lot of stem cell biologists.
when you say self-organized, like you would just give it a general direction to grow in, and then it would create a like it would create some structure of a brain that like made sense to it. Like
Yes,
I just okay,
exactly that. So the initially you're making uh, brain cells of different types. So a cell that's going to be in the front of the brain versus the back of the brain. So you make those cell types front versus back, but then they self-organize. The guys who are front of the brain end up in the front of these organoids, and the guys who are in the back migrate to end up in the back. So they actually self-organize where they should
oh
make some front of the brain neurons and some back of the brain neurons and put them together. They self-organize, and the front ones go to the front, and the back ones go to the back. And
wow,
that's sort of encouraging, right?
how is it even possible though? Like I don't like
There was
if
people
it
argue
is
they're responding to gradients, and so the default is you become anterior part of the brain, the front of the brain, and then
okay.
you specify with morphogens, with drugs, to make more and more posterior parts of the brain. And people argue these mini brains are doing that themselves. They're generating an anterior posterior axis. You don't need to do anything to the front of the brain because it's the default. But then you start using those morphogens to create the middle part, the back part, the spinal cord, what you've got. Yeah.
Interesting. Wait, and then a morphogen is what exactly?
It's a drug or a growth factor that indicates positional value, front versus back. And so if
Okay.
you experience that morphogen, you say, okay, it's often in a gradient. So it'll be low in the forebrain and high in the back brain. So depending in what part of the brain you're in, that morphogen will say, make a front of the brain cell, make a middle of the brain cell, make a caudal part of this brain cell. And so the morphogen may provide that positional information because it's highest at the back end and lowest at the front end. And so anybody who can read that gradient should be able to know what type of cells to produce.
Interesting. But like that, is that gradient alone like enough information for the stem cells to know how to make a brain? Because like in like children, like it's the whole your whole system is like developing at once, right? So there's lots of different like stimulants as to what should be growing where.
Yeah,
There's
this is something that happens before you're born. This is a really early event, right? You got it before you're born, you got to know where the front and the back are. So this
yeah.
is happening in the first few weeks after fertilization. This is a very
Okay.
early event. And sorting out front from the back is a really important thing to do.
Okay. So this
This is one of the first steps you'd have, right? Because once you
okay.
get a brain cell, then you've got to specify it positionally. Is it should be a front or should be a back? And actually, we did these experiments a long time ago where uh, we showed genes that are expressed in the middle of the brain versus the front of the brain. And if you take some of the earliest stem cells from the back of the brain that are expressing a gene that says back of the brain and stick it into the front of the brain, they'll gradually adapt to their new environment and start producing front of the brain cells.
Oh wow.
And so these
Wow.
cells are both sensitive to cues, morphogens that tell you make front versus back, but they're also generating them in some ways. Once a small number of posterior brain cells get in the front of the brain, then they receive all the front of the brain signals and they start making front of the brain and generate one of the morphogen gradients.
Interesting. So that like level of brain you're talking about, that's like that would be like the brain in an embryo that was like 3 weeks old or something, or like
Yeah,
4 weeks old.
yeah, yeah,
And
yeah.
then once
That's
you go
really,
be okay.
then things get really complicated. That's
I was
the
gonna say, yeah, like
yeah.
once you go like beyond that, like how much
Yeah,
like
because the front of the brain is really different than back of the brain, and so
yeah,
generates very different types of structures. So yes, they decided they're going to be front of the brain, but now you've got to decide on all the bits of the front of the brain, and that
yeah.
problem's not solved yet. And that's what happens for 90% of embryo development till you're born. The first 10% we've got a handle on, but after that it's pretty messy.
Okay, yeah. Because I was gonna say, like, how would you grow the other 90% in if you had a petri dish, how would you grow the other 90%? Like, is there any
Yeah, well, I mean, that's the idea of the self-organization is that if you can just create front, middle, and back, then these cells will self-organize. And the example is the one I gave you. You put some cells from the back of the brain into the front of the brain, some stem cells, and they'll take on a front of the brain phenotype. So they're responding to the new environment and becoming part of the new environment. And so the hope is that maybe if you just give them the basic instructions, front versus back, then they'll do everything else by themselves. It's partly a crazy hope because it took 40 years to figure out what the morphogens were. It's going to take even that longer to figure out how you actually self-organize a brain, how it happens.
Yeah, I'm just trying to understand, like, if you have a brain growing in the lab, how it knows by itself, like, which way to grow. Yeah, I guess like growing front and back and doing that relatively basic calculation on its own
Yeah.
seems like it's the easy part, but then how it knows what parts of the brain to develop for like different senses or like for like holding like memories. Like it
Yeah.
seems like you would there has to be more direction that you give it.
Yeah, but would think that was true, except that these little self-organized organoids seem to look like the front of the brain. They look surprisingly like the front of the brain, but they don't have all the attributes, but they have a similar look to them. So,
Wow, yeah, that's okay.
and I mentioned just front versus back, but of course there's a positional information in top versus bottom of the brain too. So there's another gradient that's high at the bottom and low at the top. And so it tells you, well, you're front bottom of the brain. So you're getting closer, right? So the more you specify all the coordinates of what you are through these gradients that go dorsal, ventral, up and down, or anterior, posterior, then at least you're in the right ballpark. You've got cells that should be in the right ballpark. Then self-organizing interactions, it's argued, allow you to generate more brain structure just without doing anything. It just self-organizes. So yeah, the simple problem that they're having a hard time solving now is once these mini organoids, brain organoids, get a little bit bigger, they start dying from the inside because all the nutrients are on the outside. And so
Oh,
it's a real problem right now because you're getting this self-organization and then the cells in the middle are dying on you. And so how to keep the cells in the middle alive and get the nutrients in. So people are now trying to grow these mini brains along with blood vessels. and the blood vessels sort of come in, and you can shoot nutrients through the blood vessels to try and keep some of the cells in the middle alive. So that's one of the things people are trying to do now.